Welcome everybody to the channel. So let's start today's topic with iron absorption. Now iron can be in two forms. It can be ferrous form and it can be in ferric form. Let's say this is food particle and this is heme from hemoglobin. This both will have ferric form. That means Fe plus 3. Now there are mainly two ways Fe plus 3 can be formed, that is from food and heme part. Both can enter the systemic circulation from intestine. So let's say these are the cells of intestine from where the ferric part will absorb in the body. Now here there is something that is called as vitamin C ferroreductase. So what happens here, the ferric from, that means Fe plus 3, will convert into Fe plus 2. It will reduce to Fe plus 2. And then with the help of a transport called divalent metallic transport 1. It's also called DMT1. So it's the trick that it's divalent, right? Di means 2. That means it allows only the iron which has plus 2 ion on it. That is ferrous ion. So now what? Ferrous will enter the intestinal cell. This we have drawn is the intestinal cell. Now the Fe2 will come inside the intestinal cell and it will convert to Fe plus 3 again. Now this stored Fe plus 3 in the cell is known as ferritin. But some Fe plus 2 also will remain. It will not convert to Fe plus 3. So let's say this is the inner layer of intestinal cell and there is ferroportin present here. This is ferroportin. What it will do? It will exit the Fe plus 2 out of intestinal cell. Now what happens? This is Fe plus 2 and there is something called as hepastine, which will convert Fe plus 2 again to Fe plus 3. Okay, now see what happens. We got Fe plus 3 in the systemic circulation. See, this is the systemic circulation. And we got Fe plus 3 molecules from hepastine. Now there are something called as transferrin in the systemic circulation which will bind to Fe plus 3. So mainly if the transferrin is in unbounded form, then it's called apotransferrin, and if it's binding with Fe plus 3, then it's called transferrin. So let's say this is apotransferrin and this is transferrin. There are two molecules of Fe plus 3 binding to transferrin. Now this is called transferrin. Now what happens? From here it will go where? So let's assume this is transferrin with two molecules of Fa plus 3. Remember, it's two molecules attached. Now this will go to either in bone marrow. This is bone marrow. Either it will go to bone marrow or it will go to liver. Now here what we know, that in bone marrow erythropoiesis occurs. So it will help in erythropoiesis, and this is the major absorption occur, while 10 to 20% will go to liver. So it is called minor, and 70% to the erythroblast. Now let's assume this is the hepatic cell. Let's zoom it out. So this is hepatic cell or liver cell. Now when the transferrin attached to Fe plus 2 ions enters the cell. So these are, we are zooming out here, right? So this is Fe plus 2 binding with transferrin. But in hepatic cell, we need to remember that it is all present under a vesicle, which is formed during the endocytosis of transferrin binding Fe into the liver cell. Now what happens here? Let's say there is H plus ion entry in this vesicle. That means increased acidic nature. This will lead to detachment of transferrin and Fe plus 2 molecules. Now they are detached, all three are detached, and they have to get out of the cell now. So they get out of the cell. So detachment occurs, and they will again enter the systemic circulation. Right? So this is the physiology. Now there is a point you need to remember in ion absorption, and it's important as for exam point of view as well as for university exams. There is something known as hepcidin. You might have heard about it. So it does what? It prevents ferroportin, responsible for spleen macrophages, and as it prevents the ferroportin, it decreases iron level in body during iron overload in body. So when there is increased iron in the body, it will block the ferroportin, and it will decrease the further absorption of iron from the intestine. Mainly there are two things which enhance the effect of hepcidin. They are interleukin 6, something called as HFA protein. Now this protein produce hepsin. So what happens? Let's talk about HFE now. It produces hepcidin, right? So now if mutation occurs in it, what happens? We have something called as hereditary hemochromatosis. Very important for exam. You need to remember, and you will only able to remember it only if you know the physiology of iron. And this will lead to Fe overload, and this can be fatal. So you need to remember it. What's the importance of HFE protein? How it is linked with hepcidin. So thank you guys. So if you like the video, please subscribe the channel, like and share this video. It will motivate me more to make such amazing videos for you. Thank you.